Hey guys, it's Mr. Burdett. Super glad to have you back in class for AP Bio, and we're just going to dive right into what we're covering today. So to help you get started, I want to show you all the materials that you'll need to be successful for today's lessons. The first thing are this video. Make sure that you have this pulled up. Second thing are your notes for today. So you're going to submit this on Classroom in this post, and I'll show you how to do that at the end in case you're unsure. But as we go through the video and I go through the slides, you just follow along and fill in the notes as we go through. The way to do that is when you open this file, you're going to go to File, make a copy, name it with your last name, so and today's lesson, right? Then you'll just click OK, and once you've done that, you can type directly into this document and you'll upload that at the end of our class session. So since you need the video and the notes, I would just split screen them. You can do that using this button here and then kind of size them however works best for you. But let's get into it. So today's agenda, we're not going to do the catalyst just because it's kind of difficult to navigate that on an online setting, and we'll start with our notes. After that, you guys will have some independent practice, and your homework is just to finish that practice. Remember, when I say homework, you're already at home. That just means that the other assignment that's due by 8 a.m. on Wednesday is the independent practice assignment. It looks like this. You can just answer these on a separate sheet of paper, and then you can upload that file to the same post on Classroom. Or you can do it in a Google Doc if that's something that you prefer. I just like to hand draw Punnett squares, which you're going to see today, because I have to try to use these tools to show you examples, so we'll see how that goes. Couple of quick announcements regarding your AP exam and College Board. So we are 11 class days away from your AP exam. Actually, I think fewer than that at this point. So I want you to make sure that every time you're attending my class online, you're giving it all of your attention. If you can find a space to devote just for classes, that'd be excellent. And I'm sure you have plenty of other AP classes to worry about, but try to get into a headspace where you're focused entirely on the class that you are in. I'm going to be posting optional work closer to deadline. It'll kind of be like a review guide to help you prepare for the AP exam if you intend to pass it. And there are a couple updates from College Board which we need to review. So the important thing is that College Board realizes that this is a time of stress and that this is kind of an unprecedented adventure for all of us. So they have removed some of the required sections, specifically our last 2 units that we were going to cover in this class. So evolution and ecology are no longer required, which means all we need to do is get through genetics, and then we can start reviewing some of the content that we know will be on the exam. Sad that I can't have one of you read these out loud for us, but here are our learning targets. So today you're going to be able to create Punnett squares for non-Mendelian inheritance. We talked about Mendelian or kind of like basic inheritance prior to our break. But now we're going to get into the nitty-gritty of what happens when things don't follow the rules. Then you guys will look at multiple alleles and polygenic traits, and then finally epistasis and lethal genes. All of these are types of Punnett squares and genetics problems that might show up on your AP exam. So let's go ahead and get into it here. Our first type of non-Mendelian inheritance that we need to talk about is incomplete dominance. Incomplete dominance is when the traits actually blend. So if you remember in the basic genetics intro that we did, we talked about how traits don't blend together, that if you have blue eyes or brown eyes, your kids will have blue or brown eyes, that it won't mix the two. But some things don't follow that rule. So the classic example that you'll see through AP are snapdragons. And these are types of flowers. Here you can see that the red, or the 2 recessives result in a white flower. 2 dominant alleles result in a red one. But if you mix those, if you have a heterozygote, then you get a pink flower instead. So we're going to do an example together, and then you have one in your notes. So make sure you're following along. So you're going to cross 2 pink flowers and see what happens for your notes. But for our example, we'll cross a pink flower and a red one. So you guys get to see my excellent artistry. These are going to be so bad. I'm so sorry. All right. So first thing we're going to do is draw a 2x2 Punnett square. Oh my God. And then you're going to add your letters. So I know I have a pink flower. Remember, pink is a heterozygote. So that's one capital R, oh Jesus, and one lowercase. Then I have a red flower, so that's two capital R's. Now on the actual, on the actual AP, remember you guys don't need to draw these if you can just do it in your head. And honestly, for our notes in class, that's fine too. But if there's a Punnett square on your note sheet, make sure that you fill that in for us. Whew, buddy, this is a struggle. So we've got the pink flower up here on the top. We've got the red one on the side. We're going to fill in this cross. So we know that these two are homozygous dominant. And these two are going to be heterozygous. So if I were interpreting these results, if this were a normal cross, we'd say that 100% of them have the dominant trait, but that's not the case anymore, right? So when we look at this, we know that 50% of the flowers are going to be red. And we know that 50%, oh my gosh, are going to be pink. Y'all, I don't know how Mr. Anderson does this, but we're going to get through it together. So go ahead and pause the video and you will fill in your notes right here the answer for what happens when you cross two pink flowers. All right. So let's go ahead and move on. I did want to show you this image just because it shows you different notations for what these would look like. So instead of big R, little r, they use R1 and R2. The reason for that is they want to show you that the traits blend, so they're incompletely dominant. So there's technically not a dominant one, even though there is a dominant trait. So it's the same process here. They used R1 to represent red, R2 for white, and then R1 and R2 together would give you a pink flower. So sometimes you'll see this with rodents as well. So if we take a black rodent and we mix it with a white one, then we get a gray. You can do this cross in your notes if you want for extra practice, but it's not required. It will help you when we get to the independent practice here. But remember, if you're crossing a black one with a white one, you will get all gray offspring. And then when you cross the F2, you're going to get 25% black, 25% white, and 50% gray. So our next example is something called codominance. So instead of things blending together, they just both show up. They are both dominant traits. If you cross a red cow with a white cow, you get something called a roan cow, which you can see right here. Roan cows are a codominant blend of both white and red. So instead of the cow being pink, you can see both white and red fur on this cow here. In your notes, you have a place for you to cross two roan cows right here. So go ahead and pause the video, see if you can do that one on your own, and then I'll go ahead and set up a solution for you to review. All right, so hopefully you solved this for yourself, but it works very similarly to how incomplete dominance works. We can see we've taken two roan cows, that's capital RW, capital RW. And when we cross them, we get these specific results. So we've got one cow that's red, two that are roan, one that is white, which you can see in my very poorly drawn distribution right here. All right, so next up in our notes, we have sickle cell and other autosomal recessive traits. The notation for sickle cell looks like this. You can leave off the HBs if you want, if that will help you to draw this and recognize it more easily. So you can just say AA for no sickle cell, AS for the sickle cell, and for the trait here, and then SS for actually having the disease. So anytime that a trait says it is autosomal recessive, which you see right here, what that means is you have to have two lowercase copies. In this case, we're using capitals, but two of those recessive copies in order for a person to have the trait. If someone has one of each, like the dominant and recessive, then they are a carrier for the trait, but they don't have the disease associated with it. So in your notes, you have a place for you to cross parents who are carriers and determine the probability of their child actually inheriting this trait. Remember, to inherit it, they have to have both lowercase copies. Let's take a second, answer that question, and then we'll go over the results. All right, hopefully you have caught on by now, but if we cross two people who are carriers, that would be like crossing one capital, one lowercase. That means 25% are going to be completely recessive. There's a 25% chance that their kid would inherit that trait. Now we're going to move on to blood types. Blood typing is interesting because it has multiple alleles. Anytime that a trait has more than two options, it's going to have these multiple alleles. So for blood type, you can be type A, type B, type AB, type O. Those are our 4 different blood types that we have. And then, of course, you can be positive or negative, but that doesn't really affect our practice that we're doing right now here. That's just the presence of an Rh factor. So you guys are going to need this table. So feel free to pause, copy down what you need. I use the notation right here, but you can use the one with the eyes as well. You could see either on the AP exam, so just be familiar with both. The thing to note is that someone who has type A blood can have 2 capital A's, or they can have a capital A and then the recessive trait, which you can denote with an O or a lowercase I. For someone to be B, they need either 2 dominant B's or B and O. If someone has A and B, those are technically codominant, so they would get type AB blood. And then for someone to have type O blood, they have to be completely recessive, so they have 2 lowercase i's or 2 lowercase o's. So knowing all of that, let's go ahead and do a practice problem. So when we're crossing blood type, the thing that you need to keep in mind, you have to know whether the type A or B person is heterozygous or homozygous. So that means we have to know whether they have 2 capital A's or capital A and a O or lowercase I, depending on how you're noting it. In your notes, you have a space to perform one practice cross for this. If you want to do all 3 of the ones from the slides, that's fine, but I wanted to make sure that this was manageable so you could move into your independent practice. So if we cross someone who's AB and AO, what is that going to look like? I'm going to give you guys some time, so pause the video here if you'd like, and then when you've finished setting it up in your notes, you can come back and watch to see what the solution looks like. All right, so here are your results. Just gonna zoom in so we can figure out how to interpret these. So if we look at these, capital A, and capital AO are both type A, which means there's a 50% chance their kid's gonna have type A blood. There's a 25% chance their kid will have type AB, and there's a 25% chance that their kid will have type B. Hopefully you guys got that. If not, feel free to go back and review information if you need to. So now let's move into polygenic traits. This is when there's a trait that's controlled by multiple alleles. This one is what we just looked at with blood types. And then there's some traits that are controlled by more than one gene, right? So in this case, we just have multiple alleles. And if we were going to figure out the genotypes for these aliens, right, you would have 4 different letters associated with them, just like we do with blood type. Our next category are polygenic traits. These are ones that are controlled by more than one gene. When we're talking about these, variation in them is continuous. Another word we use for that is that it is quantitative, so it adds up. The prime example that you'll see in humans is actually like hair color, height, skin color, anything where there's a ton of variation. It's technically a polygenic trait. When we're looking at polygenic traits, sorry, we'll go back to this slide. The thing to keep in mind is that the more capital letters it has, the more likely it is to be in that dominant spectrum. So if we look at seed color in wheat, if they have all recessive colors, they're going to be light. If they have all capital letters, they're going to be dark. If they have a mixture like these, then they're going to be in those intermediate ranges. The same with human skin tone. So you can tell by my lighting in my house that I'm very pasty, right? So I would technically have all recessive colors. Someone with very dark skin would have all capitals. Someone with lighter skin would be in this range, and so on. When we look at polygenic traits, an interesting pattern occurs here. So if we take a P generation, remember for P, we're always causing a completely dominant with a completely recessive. And if we have this parent generation here, what happens is in the cross, all of the offspring are completely heterozygous for F1. In the F2 generation, because we have all of this variation, you actually end up with 7 different phenotypes just from having 3 different letters for each parent. So again, pasty over here, very dark skin over here, and then the spectrum in between. The interesting thing with polygenic traits is that they are normally distributed, so they have a bell curve to them. That means that people, for the most part, are going to fall into this middle spectrum, the intermediate trait. The further you get to the margins, the less likely people are to have that trait, just because of the way that the population is distributed. And a good example of this would be looking at height. So this is just a bunch of random people on a football field. They had them line up by height, and you can see even with a random sample that's not representative, you can see that bell curve structure to the height, right? People are less likely to be 5 foot. People are less likely to be 6'5". People are most likely to be between 5'6" and 5'10", which would be the center of that bell curve. We have a few more categories to get through, so sorry guys, but I want to make sure that you know all of this for your exam. So our next category are sex-linked traits. Sex-linked traits operate much the same that regular traits do. The only thing is that we're also considering the sex of the organism. So in your notes, you have a place for you to do one sex-linked trait practice problem. We've got a white-eyed male and a red-eyed female. We know that eye color is sex-linked. It doesn't tell us whether it's dominant or recessive, so just assume that it is a dominant trait, meaning if they get one copy, then they have that dominant trait. I want you to try this on your own, but if you're like uncomfortable because there's extra letters, feel free to just continue watching. But if you can do it, go ahead and try it on your own and then come back after you stop pausing the video and check the answer. All right, so if you finished, you can see right here what that answer would look like. So if we're interpreting this data, all of the women are going to be heterozygous for the trait, but they do have red eyes. Remember, men, when it's a sex-linked trait, only inherit one copy because the Y chromosome doesn't have it attached. So all of the men have red eyes and they are not heterozygous. It's just completely dominant. All right, my friends, with that, we are almost through. We just have a couple more examples to go through. So the first are environmental and phenotypic traits. This is when the environment that an organism is placed in actually affects the traits that it displays. So sometimes temperature, water, food sources, and the availability of them can affect how a gene is expressed. For example, rabbits have a gene that codes for darker pigments at lower temperatures. The reason for that is, remember, darker skin can absorb more light. Sorry if you heard that. My neighbor is stampeding upstairs. So the darker skin can absorb more light. They've actually done studies with this where they tape ice packs to the rabbits, which is kind of mean, but they're fine, they have fur. And you can see the effect right here, where the skin pigmentation changes as a result of the temperature. Next up, we have epistatic alleles. We'll practice with these more closer to the exam. But the thing to note about epistatic alleles is that they have kind of like a hidden trait. So Labrador retrievers actually have 4 different alleles for coat color that result in 5 different color combinations. When we're looking at epistatic traits, there's always one that's dominant to the other. So black is our dominant trait, and it is dominant to the chocolate lab, which is a lowercase b. But yellow is the recessive epistatic trait. What that means is you have to have 2 copies of it for it to show up. But if you have 2 copies of the yellow trait, which would be 2 lowercase e's, then it actually blocks the expression of both black and chocolate. We'll practice with these, like I said, closer to the exam, but I just wanted to give you examples of those if you need them. So if we're looking at actual coat color, even though yellow is the recessive trait, it's more likely to appear than chocolate. The reason for that is that if you have any pairs of lowercase e's, then the yellow displays regardless of what's in front of it. All right, moving on to our final type are the lethal genes. This is not in your textbook, but it is sometimes on the AP exam, so I wanted to cover it. Lethal genes are only present when both alleles are present. The lethality, remember the word lethal just means deadly, can happen before birth or after. So Huntington's disease is a disease in humans caused by a lethal allele, but it actually doesn't affect you until you're in your 60s and 70s. Other examples that often show up on AP exams would be like mouse coat color and then the creeper legs in chickens, which you can see over here on the right. Another would be Manx cats, which don't have tails. The reason for that is that tails help them with balance, so it's technically a lethal gene because if they were in the wild, they wouldn't survive. This creeper allele over here in the chicken causes it to have really short legs, which means it can't navigate as freely and is more likely to be eaten by predators. So our last example before you move into your independent practice is a lethal gene with a yellow mouse and a brown one. So I want you to go ahead and do this cross. Basically, remember if it has 2 of the recessive alleles, so if we look at the Yagudi mouse over here, if we cross 2 yellow, then they're both carriers. What that means is there's a 25% chance that the lethal gene would express and it would die as an embryo. There's a 50% chance they would have yellow offspring. There's a 25% chance that one will be brown. So if you cross a yellow mouse, that's 1 dominant, 1 recessive, with a brown, which is 2 dominant, what is the probability that their offspring will die, and what is the probability that they'll be carriers? Go ahead and answer that, and then we'll come back. All right, so I used a little bit different recording or notation for this. Instead of AY, I just used a lowercase a for the recessive and a capital A for the dominant. But what you can see from this is that there is a 50% chance their offspring will be carriers, but a 0% chance that that lethal gene will express. And that, my friends, is the end of our notes for today. So the notes are pretty long, but the practice shouldn't be. Remember, you're going to upload your notes on Classroom. You're also going to answer these questions on a separate sheet. You only need to do questions 1 through 7. So don't worry about the second page. And then you're going to upload all of this to Google Classroom once you complete it. So if we go in Classroom, I'll show you what this would look like based on our teacher class that we were in to help us prep for remote learning. So I'll go over here to my classwork. When you view the assignment, there is a place over here for you to add your documents. So you're going to attach both your notes and your practice problems. You can do it as an image or you can do it as a Google Doc, whatever works best for you. And then once you've added both, you're going to click mark as done or turn in, whatever button shows up on yours, so I know that you finished that work. If any of this is confusing for you, feel free to pop into my office hours. I will be available pretty much anytime from 2:45 to 3:45 to answer any questions you might have. And I'll be on from 9 to 11 a.m. on Tuesday to help you guys with that. Bye.